Hello and good evening, everyone. I hope I'm audible. Can someone confirm?
Yeah, ma'am.
Okay. Welcome, everyone, to the 11th TA session for Drug Delivery Principles and Engineering. So, before we start the session, this session is being recorded and will be uploaded on YouTube. Also, the presentation that we'll discuss today will be uploaded on Google Drive and the link will be shared with you. So, let's start the session. I'll be sharing my screen now. Please confirm if you all can see the screen as well. Can you all see my screen?
Yes, ma'am.
Okay, so let's start. So in the previous week, these were the topics that were covered: gene delivery vectors, gene delivery polymers, genes as vaccines, and the vaccines for gene delivery and other variants, cancer vaccines, and cancer vaccine for immunotherapy. We'll start with the assignment. So here is the first question. Select the correct option with regard to vaccines for cancer. The options are: they have to be therapeutic as prophylactic cancer vaccines are unsafe and cause tumors. These vaccines are agents that can recognize and attack tumor cells. By isolating cells from autologous tumor tissue, a wide range of antigens can be presented. Adjuvants are not required for tumor vaccines, or cancer vaccines are used for training neutrophils to attack the cancer tissue, thereby annihilating them.
First option.
They have to be therapeutic as prophylactic are unsafe and cause tumors. Okay. Anyone else?
Third one, ma'am, by isolating the cells from the third one,
Okay.
I think.
Okay. What about others?
No, it is not first option, madam. Sorry, madam.
Okay. Yes, the correct answer would be the third option. And yes, therapeutic, the first option that you're saying, they have to be therapeutic as prophylactic vaccines are unsafe and cause tumors. That is not the case. Both therapeutic and prophylactic cancer vaccines exist. The third one is based on therapeutic vaccines for autologous by using autologous tumor cell vaccines, wherein they isolate cells, lyse, and inject with adjuvants. This presents an entire spectrum of antigens, but there is a slight risk of autoimmunity because here, uh, slight risk of autoimmunity, but not as compared to allogenic tumor cell vaccines. Also, it needs a lot of tumor sample and may involve some surgery. So the third option, that is by isolating cells from autologous tumor tissue, a wide range of antigens can be presented, would be the correct option. Okay, here is the next question. Therapeutic cancer vaccines are a type of active dash with the aim to produce a dash immune response to tumor antigens or tumor associated antigens that are able to dash cancer cells. I'll give you some time to go through all the options. Let me know what you think the answer is.
Ma'am, maybe second one.
Okay. Anybody else?
Maybe second option, ma'am.
Okay. Yes, second option is the correct. Immunotherapy, specific and destroy. Just now we discussed in the previous question, right? Autologous tumor cell vaccines. By isolating cells from the autologous tumor tissue, a wide range of antigens can be presented. This makes it more specific to destroy the cancer cells, and this is a type of immunotherapy. So the second option, immunotherapy, specific and destroy, will be the correct answer. Okay, let's move on to the third question. Dash polymers are generally used in gene delivery. You have to select two correct options: cationic, anionic, neutral, positively charged, negatively charged, doric, or non biodegradable.
2 and 4.
2 and 4. Okay. Anyone else?
Answer may be cationic and positively charged polymers.
Okay. What about others? Anybody else would like to answer?
That fifth option, Doric means what, ma'am?
Uh, I'm not really sure. Um,
Because I am hearing this first time.
yeah, same. I'm not sure. I have to look what is.
Yes, ma'am. Yes, ma'am.
Okay, so those who answered cationic and positively charged, it is the correct answer. Cationic polymers or positively charged polymers are basically used in gene therapy because DNA itself is very highly negatively charged polymer, so it can form complex with the positively charged polymers or the cationic polymers to form packets or particles, and these can be then endocytosed by the cells. Okay. Let's move on to the next question. Fourth question. What makes chitosan a suitable polymer as a gene delivery? You have to select two correct options. Options are positively charged, biocompatible, synthetic origin, hence less immunogenic, negatively charged, neutral charged, or non biodegradable.
Maybe first and second.
First and second, okay. Anyone else? Yes, you are correct. Chitosan is suitable as a gene carrier because it is positively charged and it is biocompatible. Uh, well, it is not synthetic origin, it is a natural polymer. So that's why it is less immunogenic. And yeah, it is positively charged and non biocompatible. So, this Here, this is from the slide itself. So cationic biopolymer that can complex with the anionic DNA. It is a natural polysaccharide, so it is non-toxic, non-immunogenic, and biocompatible, and it is suitable for mucosal delivery. Also, the available amine groups on the surface ligand attachment and potential for targeted delivery. Okay. So here is the next question. Which of the following polymers are not commonly used for gene delivery? Which of the following polymers are not commonly used for gene delivery? Polylysine, polyarginine, PAI, chitosan, or PVC?
PVC.
Okay. All of you agree with PVC? Or anyone else has any other answer? Okay. Yes, PVC is the correct answer. So polylysine, polyarginine, these are positively charged. Polyethylenamine and chitosan, polyamidoamine can also be similarly. These are also positively charged polymers that can be used for gene delivery and which are commonly used. Only PVC is the only option which is not commonly used for gene delivery. Okay, here is the next question. Which of the following is false for allogenic tumor cell vaccines? It uses few established human tumor cell lines, limitless source and cost effective, highly successful approach, risk of immunity, may not be effective at all.
Option 5.
May not be effective at all. Okay.
Option third, ma'am, highly successful approach, maybe.
Okay. What about others?
Third option, ma'am.
Third option. Okay. Okay. So here it is asking which is false for allogenic tumor cell vaccines. So yes, the third option will be correct, which is highly successful approach. This is not a very highly successful approach. It may not be effective at all. That is one of the options. So these are therapeutic vaccines, allogenic tumor cell vaccines. It uses very few established human tumor cell lines, limitless source and cost effective, risk of immunity, and may not be effective at all. Okay. So seventh question, which of these is not a potential tumor human immune evasion mechanism? Physical barriers imposed by tumor architecture, induction of host myeloid and lymphoid suppressor cells, blocking T cell effector function, resistant to apoptosis, downregulation of MHC, evasion of T cell recognition, upregulation of antigen processing.
Option 5.
Downregulation of MHC, okay. What about others?
Last two options, ma'am, I think so.
Last two options.
I think so.
Okay.
Last option.
Okay. Anyone else would like to answer?
Aviation of T cell recognition.
Okay. Okay. So here the correct option is upregulation of antigen processing. Rest of the options, if you see here, it is asking which is not a potential tumor immune evasion mechanism. So we are trying to evade the immune system. So we are trying to suppress the immune system. That is how you will be able to evade it. So here only option upregulation of antigen processing is actually enhancing the immune system. All the other options such as physical barriers imposed by the tumor architecture or induction of host myeloid and lymphoid suppressor cells, blocking T cell, resistance to apoptosis, down regulation of MHC and evasion of T cell. These are basically suppressing or down regulating or evading different immune cells. The only option is upregulating the antigen processing is basically not evading the tumor immune evasion mechanism. So this will be the correct option. Upregulation of antigen processing. And here are other potential tumor immune evasion mechanisms. These four, some of the options were already in the options. Physical barriers imposed by tumor architecture, induction of host myeloid and lymphoid suppressor cells, blocking T cell effector function, expression of proteases that interfere with cytotoxicity, expression of FASL, downregulation of cytokine receptors, resistant to apoptosis, evasion of T cell recognition, loss or mutation in target antigen, downregulation of MHC and other components important for antigen processing, expression of ligands for killer inhibitor receptors, and shedding ligands for killer activator receptors. Okay, so here is the next question. What are not the objectives of gene therapy? You have to select two correct options. Replace a missing or defective gene, repair a non-functional gene, produce therapeutic proteins that help treat a disease, vaccinate a person against an infection, cleave healthy genes already present or to generate haploid cells, cause dysfunction at a cellular level. You have to select two correct options. What are not?
Last two options.
Okay.
Yeah, option 5 and 6.
Okay. Anyone else? Yes, last two options are correct. That is, cleave healthy genes already present to generate haploid cells or cause dysfunction at a cellular level. Apart from this, all the other options are the objectives of gene therapy. That is, it to replace a missing or defective gene, to repair non-functional or malfunctional gene, produce a therapeutic protein that has to treat the disease, and can vaccinate the patient against an infection. Okay. So here is the ninth question. What property of the polymer causes proton sponge effect? Long chain length, presence of secondary and tertiary amines, spherical shape, negative surface charge.
Option two.
Okay. Anyone else?
Option 4.
Okay.
Maybe second option, ma'am.
Okay. Okay. So second option is the correct answer. Presence of secondary and tertiary amines. In the initial lectures itself, this proton sponge effect was introduced in the topics. So proton sponge effect, so particles that are uptaken through endocytosis, they get localized in the endosomes and lysosomes where there is very low pH. So it is kind of acidic environment. And to escape this, proton sponge effect is used. That is, the particles are designed to carry secondary or tertiary amines. So the pH of the acidic medium, it buffers that pH of endosomes or the lysosomes and results in proton influx through the endosomal proton pumps. So it attracts more and more ions inside the lysosomal or endosomal compartment, and this is followed by the chloride ion transport, leading to osmotic swelling and bursting of the vesicles that releases the particles out of the endosomal or lysosome into the cytoplasm. So you can use presence of secondary or you can add secondary or tertiary amine based or design particles with secondary and tertiary amine based to for this proton sponge effect to escape lysosomal or endosomal compartments. Okay. 10th question. Which of the following can be considered as an extracellular barrier for gene delivery? You have to select two correct options. Inhibition of transfection by mucus, uptake of DNA by mitochondria, degradation of DNA in plasma, lysosomal degradation in DNA, endosomal escape of DNA, degradation by intracellular enzymes, and sequestration of gene by histone proteins.
1 and 3.
1 and 3, okay. Anybody else? Which of these is an extracellular barrier for gene delivery?
One and three, ma'am.
Okay. Anybody else would like to answer? Yes, 1 and 3 are the correct options. Inhibition of transfection by mucus and degradation of DNA in plasma. So here are the main barriers in gene delivery. Extracellular barriers include degradation of the DNA in plasma, uptake of DNA by reticuloendothelial system, inability to target DNA to specific organs, large infective via the oral route except for immunization, transfection inhibited by mucus, and the intracellular barriers include endosomal escape of DNA, lysosomal degradation of the DNA, cytoplasmic stability of DNA, and translocation of DNA to the nucleus. Okay. So we are at the end of all the questions now. Uh, I'll repeat again for those who joined late. If anybody else has any doubts, we can go through those questions again. Otherwise, I'll start from the beginning again now.
Ma'am, from the beginning, please one second.
Sure. Okay, so we are repeating now from the beginning. This was the first question. You have to select the correct option with regard to vaccines for cancer. I'll give you some time to go through the options and then I'll tell you the answer. If anybody wants to answer, they can answer as well.
Option 2.
Option 2. Okay. So the correct answer is the third option. By isolating cells from autologous tumor vaccine, a wide range of antigens can be presented. So these are basically therapeutic vaccines where in autologous tumor cell vaccines, they isolate cells, lyse, and inject with adjuvants. This presents an entire spectrum of antigens and but it needs a lot of tumor sample and may involve surgery of the individual. There can also be allologous tumor cell vaccine wherein you can get the cells from a different individual and similarly use the cells to target these cancer cells. This is the second question. Therapeutic cancer vaccines are a type of active dash with the aim to produce a dash immune response to tumor antigens or tumor associated antigens that are able to dash of cancer cells. Anybody would like to answer? Okay. So therapeutic cancer vaccines are a type of immunotherapy with the aim to produce a specific immune response to tumor antigens or tumor associated antigens that are able to destroy cancer cells. So here, as I discussed before, the autologous or the allologous cell lines can be used. That is isolating cells from the same individual or a different individual that will lead to presentation of more similar antigens and more number of antigens to specifically target those cancer cells. So second option will be the correct answer. Immunotherapy, specific and destroy. Okay. Third question, DAS polymers are generally used in gene delivery. You have to select two correct options. These are the options. Let me know what you think the answer is.
Cationic and positively charged.
Okay. Anyone else would like to answer? Yes, you are correct. Cationic and positively charged. Here it is asking for gene delivery. So gene delivery, basically it is DNA that is high, which is highly negatively charged. So if we use cationic or positively charged, it can interact with these polymers to create packets or particles, and that will lead to endocytosis by the cells as well. So we prefer cationic and positively charged polymers for gene therapy. This is the fourth question. What makes chitosan a suitable polymer as a gene carrier? You have to select two correct options.
Option 1 and 2.
Okay. Anybody else would like to answer? Yes, option 1 and 2 are correct. What makes chitosan a suitable polymer? So chitosan is both positively charged, so it can form a complex with the negatively charged DNA, and it is a natural polysaccharide which makes it non-toxic, non-immunogenic, and biocompatible. And it is suitable for mucosal delivery as well. And the available anionic amine groups on the surface helps in ligand attachment, and it is potential for targeted delivery. So these all options make chitosan as a uh, gene carrier polymer. Next question. Which of the following polymers are not commonly used for gene delivery? Okay, so here the correct answer would be PVC. Apart from this, all of them are used for gene delivery. Most of them are positively charged. Polylysine or polyarginine, lysine and arginine are positively charged amino acids. So polylysine, polyarginine, also PEI and chitosan, that is polyethylenamine, ethylenimine, and chitosan are also positively charged polymers that can be used for gene delivery. Only PVC is the only polymer that cannot be used for gene delivery. So PVC would be the correct answer. Here's the next question. Which of the following is false for allogenic tumor cell vaccines? Please go through the options and let me know what you think the answer is. Which of the following is false for allogenic tumor cell vaccines?
Ma'am, first option, I think.
Okay. Okay, here it is asking which of this is false for allogenic tumor cell vaccines. So, allogenic tumor cell basically uses cells from other individuals, not from the same site or the same individual. So here they actually use established human tumor cell lines. Also, since they are using cell lines that are there is limitless source and it is cost effective, and it is a highly successful approach. This option is rather false. It is not a successful approach. There is risk of autoimmunity and it may not be effective at all. So highly successful approach will be the correct answer, which is false for allogenic tumor cell vaccines. So allogenic tumor cell vaccines are therapeutic vaccines that uses few established human tumor cell lines, limitless source and cost effective, risk of autoimmunity, and may not be effective at all. Okay. So seventh question, which of these is not a potential tumor immune evasion mechanism? Physical barriers imposed by tumor architecture, induction of host myeloid and lymphoid suppressor cells, blocking T cell effector function, resistance to apoptosis, downregulation of MHC, evasion of T cell recognition, or upregulation of antigen processing.
Last one, ma'am.
Okay. Yes, you are correct. Upregulation of antigen processing. So upregulation of antigen processing is, this actually means you are enhancing the immune response. So this is not a potential tumor immune evasion mechanism. Rest of the options are immune evasion mechanisms. So upregulation of antigen processing will be the correct answer. And these are all the tumor immune evasion mechanisms: physical barriers imposed by tumor architecture, induction of host myeloid and lymphoid suppressor cells, blocking T cell effector function, expression of proteases that interfere with cytotoxicity, expression of FASL, downregulation of cytokine receptors, resistance to apoptosis, evasion of T cell recognition, loss or mutation in target antigen, downregulation of MHC and other components important for antigen processing, expression of ligands for killer inhibitor receptors, and shedding ligands for killer activator receptors. Okay, here is the next question. Eighth question. What are not the objectives of gene therapy? You have to select two correct options. Let me know what you think the answer is.
Ma'am, one, vaccinate a person against an infection
Okay.
and causes dysfunction at a cellular level.
Okay. Anybody else? Okay. So here it is asking what are not the objectives of gene therapy. So let's go through the options one by one. Replace a missing or defective gene. This is one of the objectives of gene therapy. Repair a non-functional gene. This is also objective. Produce therapeutic proteins that help treat a disease. Vaccinate a person against an infection. These are also the objectives of gene therapy. So cleave healthy genes and already present to generate haploid cells. This is not an objective. Further, it is not used in healthy when healthy genes are already present, and it does not. We don't need it to cleave the healthy gene to generate haploid cells. Also, it should not cause dysfunction at a cellular level. We don't want these options when performing gene therapy. So these two options are not the objectives of gene therapy. Cleave healthy genes already present to generate haploid cells and cause dysfunction at cellular level. The rest of the all four options are the objectives of gene therapy. Okay. Next question. What property of the polymer causes proton sponge effect? Long chain length, presence of secondary and tertiary amines, spherical shape, negative surface charge.
Second option, ma'am.
Okay. Yes, second option is the correct answer. So proton sponge effect is basically an escape mechanism. So when particles are uptaken by the cells through endocytosis, they get localized in the endosomes or lysosomes where the pH is very low. And so to evade this from these compartments to outside into the cytoplasm, proton sponge effect is used, wherein particles are designed to carry secondary or tertiary amines. These then can lead to buffering the pH of the endosomes, resulting in proton influx through the endosomal proton pumps, and that is followed by chloride ion transport, and this leads to osmotic swelling and bursting of these vesicles to release the particles outside. So presence of secondary and tertiary amines will be the correct option. Okay, here is the 10th question or the last question. Which of the following can be considered as an extracellular barrier for gene delivery? You have to select two correct options. Let me know what you think are the correct options are.
A degeneration of DNA in plasma.
Degradation of, okay. One more.
Ma'am, I think first option.
Okay. Yes, you are correct. The inhibition of transfection by mucus and degradation of DNA in plasma, the first and third option, are the correct answers. These can be considered as an extracellular barrier for gene delivery. So here are the barriers for gene delivery, the extracellular and intracellular barriers. So the extracellular barriers include degradation of the DNA in the plasma, uptake of DNA by reticuloendothelial system, inability to target DNA to specific organs, largely ineffective via the oral route except for immunization, and transfection by inhibited by mucus. And the intracellular barriers include endosomal escape of DNA, lysosomal degradation of DNA, cytoplasmic stability of DNA, translocation of DNA to the nucleus. Okay, we are we have discussed all the questions. Anybody wants me to discuss any question again?
Ma'am, can you repeat the very first four questions, please?
Okay, this was the first question. Select the correct option with regard to vaccines for cancer. So here the correct option would be by isolating cells from autologous tumor tissue, a wide range of antigens can be presented. So this is the type of therapeutic vaccine where autologous tumor cell line vaccines are used. So they isolate lyse and inject with isolate cells from the same uh, tumor tissue, lyse and inject it with adjuvants. This presents an entire spectrum of antigens, but it needs a lot of tumor sample and may involve surgery. Also, there is risk of autoimmunity. And this was the second question. Therapeutic cancer vaccines are a type of active dash with the aim to produce a dash immune response to tumor antigens or tumor associated antigens that are able to dash cancer cells. So here the correct option would be the second option. So therapeutic cancer vaccines are a type of immunotherapy with the aim to produce a specific immune response to tumor antigens or tumor associated antigens that are able to destroy the cancer cells. So the second option would be the correct answer. This was the third question. Dash polymers are generally used in gene delivery. You have to select two correct options. So the polymers, the kind of polymers that are used in gene delivery are basically cationic or positively charged. So these two will be the correct options. Because cationic or positively charged polymers can interact with the DNA, which is highly negatively charged, to form complexes, and also it can be endocytosed by cells. So we need it to be cationic and positively charged. Here's the fourth question. What makes chitosan a suitable polymer as a gene carrier? So we discussed before, we want it to be positively charged. Since chitosan is suitable for gene delivery, so positively charged will be one of the correct options. Also, chitosan is a natural polymer and it is biocompatible. So these two options will be the correct answers, positively charged and biocompatible, which makes it suitable for gene delivery, and it is also suitable for mucosal delivery. Further, it has free amine groups on the surface which can help in ligand attachment, potential for targeted delivery of drug. Okay, I hope I discussed all the questions.
Yes, ma'am. Thank you so much, ma'am.
You're welcome. Do you have any other doubt or any other question you want me to discuss?
No, ma'am.
Okay. Then shall we close this session?